Good morning, everyone, and thank you for the organizers to giving me the opportunity to present my work. And today I would like to talk about the mechanisms of PIK3CA-driven lymphatic malformations and the lessons we have learned from mouse models. So as was highlighted yesterday as well, there is this spectra of rare diseases, complex lymphatic anomalies, such as GLA, Gorham-Stout, KLA, and CCLA. However, there is also different lymphatic anomalies, which are cystic lymphatic malformations. Those are can be found in two distinct forms, either microcystic lymphatic malformations, which are large fluid-filled cysts often located in the head and neck region, and microcystic lymphatic malformations, which are small lymphatic channels, which can infiltrate deep in the tissue, making them hard to treat. Genetic approaches in, let's say, the past 2 decades have revealed that lymphatic malformations, 80% are caused by mutations within the PIK3CA gene, and that the mutation are found within the lymphatic endothelium itself. So it was already highlighted yesterday. So PIK3CA encodes for the catalytic subunit of the PI3 enzyme, which can be found downstream of many receptor tyrosine kinases. We have decided to focus our efforts on one of the common hotspot mutations, H1047R, which is located in the kinase domain of the protein and causes ectopic activation of the protein. Our lab has developed in the past 2 mouse models to model lymphatic malformations. One microcystic lymphatic malformation model, where we induce the PIK3CA mutation, specifically in the lymphatic endothelium, by crossing this mouse to lymphatic specific VEGF receptor 3 Cree driver. And if we induce these animals at early embryonic stages by administering tamoxifen to the pregnant dam at, for example, E11, we observe microcystic lymphatic malformations in the skin of these mice, here analyzed at E17. However, interestingly enough, there is a completely different response if we induce these animals at postnatal or adult stages. By giving either tamoxifen here at P3, we can see that the lymphatic endothelium in the skin responds by hypersprouting, more reminiscent of a microcystic characteristics. For my presentation today, I will mainly focus on our work in microcystic lymphatic malformations. So the mouse model we have developed is a progressive mouse model where we can see an aggravation of the phenotype over time. So if we induce the animals at 3 weeks of age and we analyze 2 weeks later, we can see these ectopic sprouts coming from the lymphatic endothelium, here analyzed in the dermal skin of the ear. This phenotype progressively worsens, and at 9 weeks after induction, you can see that pretty much the entire lymphatic vasculature is severely disrupted. To understand the mechanisms which underlie these microcystic lymphatic malformations, we have performed single-cell transcriptomics, where we have isolated lymphatic endothelial cells from 3 control mice and 5 mice where we have induced the mutation. We have sorted LECs based on FOX markers, and we've performed a SmartSeq2 pipeline. This is a single-cell sequencing approach, and for those not so familiar, so each cell in these graphs represent 1 cell, and the closer 2 dots or cells lie apart, they probably share a similar gene transcription profile and identity. The other way around, if cells lie very far apart in these graphs, they're molecularly or transcriptionally very distinct. So we could find 7 clusters of lymphatic endothelial cells, and based on known markers, we could already easily annotate 3 of them. Here in red, for example, capillary lymphatic endothelial cells, which can be found at the terminal ends, but also collecting lymphatic vessels and lymphatic endothelial cells from the lymphatic valves. However, we could also observe 3 additional clusters, which we did not know the identity of. All these clusters were characterized by a high expression of a protein called pentaxin 3 or PTX3, and we decided to look further. What was interesting to note is if we specifically looked at which cells came from which sample, we could see that these cells mainly came from mutant mice. So on the left, you can here see the cells coming from control animals, and on the right, cells coming from mutant animals. And you can see that these bottom clusters are mainly made out of cells coming from mutant animals, whereby red dots are cells where we could also observe the mutant transcript. This was also, once we looked at the ratios of these PTX3 high clusters, we could see that they were overrepresented specifically in mutant animals, and thus probably had expanded over the course of the development of these lymphatic malformations. So where were these cells located? So we stained for PTX3 protein in wild type samples, and we could observe staining at the terminal ends of lymphatic capillaries. Interestingly enough, if we stained mutant animals, we could see indeed this high upregulation of PTX3 in the ectopic sprouts, which give rise to these lymphatic malformations. This was also true if we looked at patient samples. So on the left, you see a control skin biopsy, and on the left, we see a biopsy obtained from a patient with lymphatic malformations, where we have identified it was a PIK3CA H1047R mutation. We decided to look deeper into which genes these PTX3 clusters express, and we could see that these clusters actually had strong signatures of immune modulation and immune regulation. This was also true if we looked at GO-term analysis, where we identify which pathways are actually enriched in these clusters. We could find many pathways related to immune regulation. One gene stood out, which is CCL2, which was, as you can see here at the bottom, was barely expressed in control cells or not expressed in control cells, but was the expression was upregulated specifically in these PTX3 high clusters. So we performed staining of CCL2, and I should mention, so CCL2 is one of the main chemokine attractants for cells of the myeloid lineage, such as macrophages and dendritic cells. Again, in control samples, we could observe no staining for CCL2 protein. However, in ectopic sprouts in microcystic lymphatic malformations, we could see that CCL2 protein was expressed. This was also true on a transcript level when we looked at RNA levels, specifically isolated from LECs of microcystic lymphatic malformations. CCL2 recruits macrophages, so we therefore analyzed the immune infiltrate in these skin lesions, and indeed we could observe that specifically in mutant animals we had a higher frequency of both macrophages and dendritic cells, but not of monocytes. Macrophages have been known to produce one of the main lymphangiogenic ligands, VEGF-C, especially in inflammation and wound repair situations. We therefore isolated macrophages and checked for VEGF-C transcript levels, and we could see that macrophages, specifically in mutant animals, produce high levels of VEGF-C, which boiled it all down to a model we had in mind where we have the lymphatic endothelium, which due to the PIK3CA mutation is undergoing hyperproliferation and migration. But also we have this unique population of PTX3 high cells at the terminal ends which expand and produce CCL2. These cells subsequently recruit macrophages which secrete VEGF-C, effectively creating a feedback loop which sustains growth of the lesion. We know that, for example, rapamycin is very good, or sirolimus is very good in inhibiting proliferation and migration, but we thought that there might be a new therapeutic window inhibiting this paracrine signaling. We therefore first assessed if we could inhibit, for example, VEGF-C. To inhibit VEGF-C, we have used an adeno-associated virus, which encodes for a soluble VEGF receptor 3 receptor, which traps VEGF-C ligands in the interstitial space. Which is often referred to as a VEGF-C trap. We induced the animals at 3 weeks of age and injected the virus at 4 weeks and followed these animals over 5, 6, 7, and 8 weeks. We could observe that indeed trapping VEGF-C in the interstitial space limited lymphatic malformation growth over all time points analyzed. We also decided to look if we could inhibit this paracrine signaling with macrophages, either by inhibiting their recruitment using an anti-CCL2 antibody or by depleting macrophages using anti-CSF1R antibody or a general inhibition of inflammation using the COX-2 specific inhibitor celecoxib. Today, I would just like to show our data on depleting and inhibiting macrophages. So to deplete macrophages, we induced animals at 3 weeks of age with tamoxifen to induce the recombination and start the process of lymphatic malformation growth. And then we administered anti-CSF1R antibody every 4th day and analyzed the animals 4 weeks later. Depleting macrophages effectively limited lymphatic malformation expansion in all the animals analyzed. We also decided to look if a general inhibition of the inflammatory response would also cause a better outcome for these mice, as of course macrophage depletion is not clinically a feasible way to go. We therefore administered celecoxib 1 week after inducing the mutation every 2nd day via oral gavage, and again analyzed the animals 4 weeks later. Again, even a general inhibition of inflammation could reduce the phenotype, and we could observe that there was a reduction in lymphatic expansion, specifically in animals treated with celecoxib, but not in vehicle controls. This brings me down to our model, where we have shown that there is a unique population of PTX3 high lymphatic endothelial cells located at the capillary ends. These cells have an immune interacting signature, and PIK3CA mutation specifically expands this population. These cells and rapamycin can be an effective target to indeed prevent this early stage of proliferation. However, these cells undergo paracrine signaling, where they upregulate chemokines such as CCL2, which will recruit macrophages, which subsequently produce VEGF-C, kickstarting the whole process and leading to a sustained growth of lymphatic malformations. I've also shown you that inhibiting this paracrine signaling, either by macrophage depletion or by a general inhibition of inflammation, is now a feasible new target. And then I would just like to end with some new data we have obtained in the lab. I've shown you before that we have this macrocystic lymphatic malformation model. However, from a practical point of view, it's not so interesting to work with embryos, as you always need to kill the pregnant dams. We decided to look more in depth into organs of mice which were bred to lymphatic endothelial specific Cre, and we observed actually this in both PROX1 and VEGF receptor 3 models, that in internal organs we could find macrocystic lymphatic malformations, for example here in the hind limb collecting lymphatics or in the mesentery. These macrocystic lymphatic malformations could be filled with chyle, or even in certain instances have penetrated blood vessels and were filled with blood. We confirmed the lymphatic identity of these macrocystic lymphatic malformations by staining, for example, for PROX1 or VEGF receptor 3. And high volume imaging also revealed that these malformations were indeed macrocystic with large lumens. We are now very much interested in finding out how these arise. Because what is interesting, in exactly these same animals, we see a microcystic response, either in the dermal vasculature, but it's not restricted to the skin, as for example, the diaphragm also shows a hypersprouting phenotype. This suggests that the same mutation targeted in different organs, so a different micro environments, has a very different effect on how these cells behave. With this, I would like to end, and I would like to acknowledge all members from the lab, and especially Milena, Marla, and Ines, who did the bunch of the work. And then our collaborators, Mariona and Euleala from Barcelona. And also the ViaCure network, which was focused on vascular anomalies and which funded me for 3 years of my PhD. And I would gladly take any questions.